Welcome Biology 400 students, this is Mr. Gales. I'm bringing you Screencast Session 1 for Central Dogma. This screencast will focus on the history of genetic research. You can see here there are 6 learning targets from your unit learning target list that will apply to this screencast. You also see that there are some related web links and activities that will help reinforce your understanding of this material. Now before we begin, make sure that you have your new screencast note-taking graphic organizer and that you've taken the time to look ahead at the main ideas and that you've recorded some prior knowledge that you may have or some questions that you may have if you're not familiar with some of the concepts. Once you've done that, we're ready to get started, so I'm going to go ahead and go to the first slide where we look at the birth of genetics as a discipline within biology. Later in the year, we're going to study Mendel's laws of heredity, what we call Mendelian genetics. Mendelian genetics is also known as classical genetics because these are the classic laws that explain how inheritance works. Much more on Mendel later, but at this point I just want to share with you the idea that Gregor Mendel, who was a simple monk working in a monastery in what is now the Czech Republic, working with pea plants, was able to work out all of the basic laws that describe how traits are passed from one generation to the next. His experiments were simple, they were elegant, and they provided 3 very important laws that described heredity. Those were the laws of inheritance, the laws of independent assortment, and the laws of segregation. And these 3 laws taken together are described as Mendel's laws of heredity. Now his work was published in the middle 1800s, and his work was not widely known. It wasn't something that would have hit the bestsellers list. This was really limited to people that were interested in heredity. It was probably primarily in the purview of scientists at the time, and it was published in German, and it was probably not very well widespread outside of the German and Austro-Hungarian empires at that point in time. Now, just because his laws were not widely disseminated doesn't mean that weren't people interested in learning about heredity. It's just that there wasn't a major focus on this particular branch in biology yet. Now, what happened because of his publication and because of the publication a few years later of Darwin's theory of natural selection describing how evolution occurs, there became more and more interest in figuring out what were the molecules, what were the factors that passed on characteristics from one generation to the next. And this begins the search for the genetic material. Now, as Mendel published his work and people began thinking about what genetic material would be and what the genetic molecule might be, they began thinking about what would be required of the genetic material. And really there are 3 things, 3 requirements the genetic material, whatever that genetic material is, that they would have to meet. And first of all, they would have to be able to encode for the production of traits. Now Mendel knew nothing about DNA. DNA hadn't even been discovered when Mendel was doing his work. And when he devised his laws, he referred to the characteristics, the material that would pass on characteristics, he referred to them as factors. So here we would have genetic factors. Mendel was able to figure out that there were factors that would eventually lead to the production of a particular trait. In this case, the trait is flower color. So there would be a large B factor which would code for or encode the production of this purple color, and there would be a small b factor that would encode for this white color. So he knew that there was some sort of factor that did the encoding that would lead to these traits. He just didn't understand what it was. So that obviously whatever the genetic material is going to be has to be able to encode some sort of information in it. Now the other thing that genetic material has to be able to do is it has to be able to replicate itself. For thousands of years, people have understood that when a living thing reproduces, it reproduces another living thing like itself. And we know that from cell theory, all living cells come from other cells. It would make sense that if the genetic molecule or the genetic material is the material that's passed from generation to generation, that material would have to replicate itself as well. We don't see zebras having dogs as their offspring. So they have to be able to pass on that genetic information not only to the cells within their bodies, but to the next generation of cells as well. Finally, genetic material has to be able to change in a controlled way. Think about this. If the genetic material for all living things was exactly the same and it never changed, what would we all look like? we'd all look pretty much the same, right? And we know that that's not the case. So the genetic material has to be able to change in a controlled way. And now, with our modern understanding of genetics, we know that what that really means is mutation. The picture that you're seeing here is of a bedbug. This is a strain of bedbug that is referred to as the Harlan strain of bedbugs. And what's unique about these guys is that they are, when they're first hatched from eggs, they are susceptible to all kinds of different insecticides, but they mutate very quickly to obtain resistance, so that in subsequent generations they're resistant to all different kinds of insecticides. And what we know is that their genes are changing very rapidly, not within one individual bedbug, but within generations of bedbugs. So what we know is that the genes are changing to allow them to survive. This change in a controlled way is really important. Like I said earlier, if genes never changed, or if the genetic material never changed, everything would look the same. And additionally, if there was some sort of environmental factor or disease that entered a population and there was no differences between the organisms in that population, it really could affect all of them. It could wipe them out. If there are small differences, if that genetic material is changed even just a little bit, then it provides some natural protection against those kinds of diseases, for instance, or chemicals. Okay, so what we've got so far is Mendel identifying the laws of heredity, and we've got scientists thinking about what the genetic material would have to be. The ball really gets rolling in 1868 with the discovery of DNA. DNA was discovered by a Prussian army doctor by the name of Friedrich Miescher, and you see the picture down here. Miescher was working as with the Prussian army, and he was actually stationed at an old medieval castle. His research lab, if you look in the picture down here, was actually the kitchen of this old medieval castle, and he didn't have very great equipment. But what he was doing was really interesting. He was taking the bandages from wounded soldiers, and he was isolating the white blood cells from the pus in those bandages. And what he found in the white blood cells was this chemical that contained a 5-carbon sugar, and it contained phosphate groups, and it contained lots of nitrogen. And what he described that, or what he named that, was deoxyribonucleic acid, or DNA. So he's the one who discovers this molecule. So again, the search for the genetic material is sort of ongoing, and the discovery of DNA in 1868 was a point in that search, or was an event in that search, but really nobody knew what it was all about. Even Miescher himself didn't think that it was related to heredity. He thought it was a storehouse for excess phosphate that the cell would use to drive cellular respiration. The next major event in the search for genetic material came in 1915 with the publication of something called the chromosome theory of heredity. Now, T.H. Morgan was a fairly well-known scientist at the time, working on heredity, working on cell biology, and he was studying fruit fly heredity. Fruit flies are these tiny little flies that if you leave fruit on your kitchen counter too long, you'll see a whole bunch of them flying around. They're really great research subjects because they reproduce very quickly, and they have characteristics which are very easy to determine. They have either red eyes or white eyes. They have long wings or short wings. So they're a great research model. What Morgan was able to do in his chromosome theory of heredity was essentially prove scientifically, experimentally, that the hereditary material was carried by chromosomes. He was able to show that chromosomes, when they move during cell division, or when they're passed apart during the division of cells, that they behave essentially like Mendel predicted they would. Mendel talked about the factors that are the genetic material. These genetic factors would separate during the formation of gametes. Well, what Morgan showed was that was actually what was occurring with the chromosomes. When gametes or sex cells would form, that the chromosomes would separate during that process. So chromosome theory of heredity was really important simply because there was a clear distinction made between chromosomes and what Mendel's laws of genetics predicted. And so now there's this, great search on for the genetic material, and all of a sudden we're pinpointing that search down to chromosomes. We have, if this was a court of law, we would have our suspect, right? We are going to now suspect that the genetic material has to be housed on the chromosomes because of the way they behave. So chromosomes would be our next main idea. The picture that you see here is a picture of chromosomes, and again, this is based on our modern understanding of it, but even back in the 1915 era when chromosome theory of heredity was published in the early 1900s, scientists knew that chromosomes were composed of DNA and protein. They didn't necessarily know the structures of those molecules, but they knew that there was DNA and protein in the chromosomes. Now, if you're limiting yourself to just chromosomes as being the suspect, and chromosomes are made up of DNA or protein, then it really is either DNA or protein, which is the genetic material. Most scientists during this time believed that it was more likely that protein would be the genetic material, and there were really two major reasons for that. When we look at a chromosome, chromosomes, even though DNA now in our modern biased opinion, DNA is obviously important, chromosomes are made up of approximately 60% protein and only about 40% DNA. Protein appears to be more important, and so that was one reason why scientists thought that it was more likely that protein was the genetic material. Now, the other major reason, when we think about the subunits, the building blocks of these molecules, DNA has only 4 building blocks. They have 4 different kinds of nucleotides based on the nitrogenous bases that are attached. You'll remember this from organic chemistry. There are thymine and cytosine pyrimidines and adenine and guanine purines. 4 different types of subunits, whereas protein has 20 different types of subunits. These are the amino acids. So again, it seems like protein is more complex. It can probably encode more information, and that's a requirement of the genetic material. So at this point, scientists are thinking it's got to be protein. Well, the next major milestone in the search for the genetic material occurs in 1928 with a scientist named Frederick Griffith. Griffith is actually a surgeon with the U.S. Army, and his experiences during World War I really shaped the research that he did. He dealt with many wounded soldiers during World War I, and actually most of the soldiers that he treated that died didn't die of their initial wounds. They died of the infections that were caused by their wounds, or that festered in their wounds. So what Griffith did was researched the pneumonia bacteria, and what he determined was that there was some factor that would cause avirulent bacteria to become virulent. Avirulent means non-disease causing, virulent means disease causing. So what Griffith was discovered through his research was that there was some unknown factor that caused what he referred to as transformation. Transformation essentially means the conversion of the genetic characteristics of an organism from one type to another. So if you think about what Frederick Griffith did, he worked with 2 different strains of bacteria, and the picture here that you're seeing, I'm going to blow this up so you can see it in a larger view. He works with 2 different strains of bacteria. He works with what is called the R strain of the bacteria, which is non-virulent, and the smooth strain, or the S strain, which is virulent. Now, if you injected living R strain bacteria into a mouse, the mouse is fine. It's not going to cause disease. It's non-virulent or avirulent. Griffith then injected the living S strain, the virulent kind, into the mouse, and the mouse dies. And not only does the mouse die, but when the mouse was necropsy, that's an autopsy on the animal, when they cut the mouse open, they found that there was all kinds of this S strain bacteria in it. So Griffith then did the next part of his experiment was to heat kill this S strain bacteria. Right? This is the S strain bacteria, but it's been heat killed. Some sort of a heat is applied to it, kills off those bacteria. He injects those heat-killed non-virulent bacteria into the mouse, and the mouse still survives. It's okay now. Right? So the big difference between these two is it's the same bacteria. This one has just been killed off with heat. Griffith thought that was really interesting. So the next step that he did was he took living R strain bacteria. Remember, the R strain bacteria are the kind that do not cause the mice to die. He takes those living R strain bacteria and he mixes them with heat-killed non-virulent bacteria, also did not cause the mouse to die, right? When he mixes them together and injects them into the mouse dies. Not only that, but when they opened up the mouse in the necropsy, they found that there were living S strain bacteria present. So really what's occurring here is something caused the transformation of the living non-virulent bacteria into living virulent bacteria. And again, he refers to this as transformation. Now, he was never able to figure out what actually caused transformation, because shortly after this research was done, Adolf Hitler came to power and he was called back into the military to serve as the U.S. prepared for World War II. Now, his research was not lost. The next group of researchers that kind of jumped onto the search for the genetic material were a group called Avery, McCarty, and McLeod. This was three researchers who in 1944 did a very important experiment that identified the transforming factor. What they did is they used enzymes to break down the different cellular components of cells that had gone through transformation. And they found that when they used these enzymes, the only kind of enzyme that would stop transformation was an enzyme called DNase, or deoxyribonuclease. This is an enzyme, as the name would imply, that breaks down DNA. Avery, McCarty, and McLeod found that this enzyme would stop transformation from occurring, and therefore they determined that DNA was the transforming factor. So they're one step closer to figuring out that DNA is in fact the genetic material. And if we look at this picture in a little bit more detail, you'll be able to see what happened. All right, so there are these heat-killed cells. Remember, the heat-killed cells don't cause the disease. What they did is they suspended the heat-killed cells into a solution inside a test tube, and they removed the lipids and the carbohydrates because, remember, we knew that the chromosomes were involved in heredity. That meant that it had to be either the proteins or the DNA, the nucleic acids, that are involved in heredity. So they removed the lipids and the carbohydrates. DNA, RNA, and proteins remain. So what they do is they add proteinases. These are enzymes that destroy proteins. The sample wouldn't contain any protein then. And so when they add the living R cells, what happened was transformation still happened. So they know that without proteins, transformation still happened. That told them that protein was probably not the transforming factor. Then they added RNase, ribonuclease. The sample should contain no RNA. They add R cells, and transformation still occurs. If transformation still occurs without RNA, then we know that RNA cannot be the transforming factor. Finally, what they did is they added their DNase to this sample. So now the sample contains no DNA. When they added the R cells to that solution, no transformation happened. So again, the conclusion that they draw, transformation cannot occur unless DNA is present, and therefore DNA has to be the hereditary material. Now at this point, you would think that this is pretty solid evidence that DNA is the genetic material. We know it's either protein or DNA, and DNA seems to be in control of transformation. But there were still scientists that were skeptical. They really thought that protein was it. There was one final experiment that was conducted in 1952 by a research pair called Hershey and Chase. What they did was actually a very elegant experiment that involved a type of virus called a bacteriophage. And you can see the picture of the bacteriophages here. A bacteriophage is a virus that literally lives off of bacteria. The word bacteria refers to the bacteria. Phage means to consume or to eat. So these are bacteria-eating viruses, essentially. Now, what's unique about viruses as a research subject is a virus is simply composed of a protein outer core or a protein outer covering and a nucleic acid core, mostly DNA, sometimes RNA. So a virus is made up of the two exact things that scientists knew were involved in being the genetic material. The research that they did led them to conclude that the active component of the bacteriophage that transmitted the infective characteristic was, in fact, DNA. In other words, if you allowed these viruses to infect a bacterial cell, that it was the DNA that was causing the infection and not the protein. So ultimately in 1952, Hershey and Chase were able to draw a very clear correlation between DNA and the genetic information by doing their experiment. Now I'm going to take a moment just to show you the experiment that they did here. This is referred to as the blender experiment. It's a very famous experiment in the history of genetic research. What they did is they mixed radioactively labeled viruses with the bacteria. These viruses were labeled either with radioactive phosphorus, which would be found in the DNA, or radioactive sulfur, which would be found in the protein. Okay, they mixed the phages then with the bacterial cells and allow for the infection to occur. They agitated the mixture in a blender, literally in a kitchen blender, to separate the phages outside the bacteria from the cells and their contents. Then they used a centrifuge to mix that all up, and basically what it did is it separated the contents of that solution now by density. They had the fluid at the top, which was called the supernatant, and the pellet at the bottom, which would be composed of the bacterial cells. When they looked at the supertinent, sorry about that, when they looked at the supertinent, what they found was that in the supertinent they found the radioactively labeled phosphorus. When they looked at the pellet, which contained the bacterial cells that had been infected, they found the radioactive phosphorus. So that, again, it concluded for them that it was the DNA that was getting into the cell and taking over the cellular mechanisms of reproduction. In other words, DNA has to be the genetic material. So from Gregor Mendel in the early 1800s all the way through to into the early 1950s, the search for the genetic material was long and ongoing, and it was this Hershey-Chase experiment that really definitively settled the question. In Screencast session number 2, Mr. Workman is going to talk with you guys about the structure of DNA and about some of the scientists that were responsible for piecing together that structure. So for next time, we'll see you in biology.